Chapter 12. The Reproductive System. Introduction. At its most basic level, reproduction is the process by which a single cell duplicates its genetic material. This is the process of mitosis. Mitosis allows us to grow and repair damaged or old tissues. In this sense, cellular reproduction enables us to maintain ourselves. However, reproduction is also the process by which our genetic material is passed on from one generation to the next. This process requires a special kind of cellular reproduction that produces special cells, the sperm from the male and the egg from the female. These join in the process of fertilization to produce a fertilized egg, or zygote. The special type of cellular division that produces the sex cells is called meiosis. Meiosis is a reduction division of the genetic material. This results in an egg carrying 23 chromosomes and the sperm carrying 23. When fertilization occurs, the resulting zygote will possess the full complement of 46 chromosomes. This chapter discusses the organs of the reproductive system that produce the sex cells, transport and nurture their development. Once an egg is fertilized by a sperm, the resulting zygote will develop into an embryo in the uterus of the female and grow by the process of mitosis into a fetus. The fetus will continue to develop until birth. The purpose of the reproductive system is to produce offspring and ensure the perpetuation of the human species. The male reproductive system. The primary sex organs of the male reproductive system are the testes or male gonads. These organs produce sperm and the male sex hormones. There are also accessory organs, like the scrotum, that support the testes. Other accessory structures nurture the developing sperm cells, and various ducts store or transport the sperm to the exterior or into the female reproductive tract. Accessory glands have secretions that make up the semen. The transporting and supporting structure is the penis. As you can see, this is the organs and ducts of the male reproductive system. The scrotum. The scrotum is an outpouching of the abdominal wall. It consists of loose skin and superficial fascia. It is the supporting structure of the testes. Externally, it appears as a single pouch of skin separated into lateral portions by a median ridge known as the raphe. Internally, it is separated into two sacs by a septum. Each sac contains a single testis. The testes plural produce the sperm and the male sex hormones. Because sperm and hormone production and survival require a temperature lower than normal body temperature, the scrotum lies outside the body cavity. Its environment is about 3 degrees Fahrenheit below body temperature. Exposure to cold, as in winter, causes contraction of the smooth muscle fibers, moving the testes closer to the pelvic cavity so they can absorb more body heat. The whole scrotal sac contracts, and a muscle, the cremaster muscle, located in the spermatic cord, elevates the testes. Exposure to heat, as on a hot summer day, reverses the process, and the scrotal sac hangs well below the pelvic cavity to avoid body warmth. The testes. The testes are paired oval glands measuring about 5 cm 2 inches in length and 2.5 cm 1 inch in diameter. They are covered by a dense layer of white fibrous connective tissue called the tunica albuginea that extends inward and divides each testis into a number of smaller, internal compartments known as lobules. Each lobule contains 1 to 3 tightly coiled tubules called the convoluted seminiferous tubules. These seminiferous tubules actually produce the sperm by a process called spermatogenesis. Spermatogenesis begins in the seminiferous tubules, as the most immature sperm cells called spermatogonia begin to divide by mitosis to produce daughter cells called primary spermatocytes. The primary spermatocytes undergo the cellular reduction division called meiosis. After the first meiotic division, the primary spermatocytes become secondary spermatocytes and their genetic information has been reduced in half. Chromosomes are reduced from 46 to 23. These secondary spermatocytes now undergo the second meiotic division and become spermatids. These spermatids will eventually mature into sperm cells. Found among the developing sperm cells in the seminiferous tubules are the Sertoli cells. They produce secretions that supply nutrients for the developing sperm cells, or spermatozoa. In the lobules of the testes, between the seminiferous tubules in the soft connective tissue are clusters of interstitial cells of Leydig. The interstitial cells of Leydig produce the male sex hormone testosterone. Thus, we see that two different areas of the testes produce two different products through two different groups of cells. The primary spermatocytes in the seminiferous tubules produce sperm, and the interstitial cells of Leydig in the tissue of the lobules around the tubules produce testosterone. Because the testes produce both sperm and testosterone, they are both exocrine glands with ducts and endocrine without ducts glands. As you can see, this is the anatomy of a testis. And this is the process of spermatogenesis, which occurs in the seminiferous tubule. The anatomy of the spermatozoa. The spermatozoa, or mature sperm cells, are produced at a rate of about 300 million per day. Once ejaculated, they have a life expectancy of about 48 hours in the female reproductive tract. They will not survive very long at all outside the female reproductive tract in the external environment. They are highly adapted for reaching and penetrating a female egg or ovum. Each sperm is composed of a head, a middle piece, and a tail. The head, which developed from the nucleus of a spermatid cell, contains the genetic material and an acrosome. The acrosome contains enzymes that aid the sperm cell in penetrating the covering of the female egg cell or ovum. The rest of the sperm cell develops from the cytoplasm of the spermatid cell. The middle piece or collar contains numerous mitochondria, which produce the high energy molecule adenosine triphosphate ADP that provides the energy for locomotion. The tail of the sperm cell is a typical flagellum. The flagellum beats from the energy of the ADP molecule and propels the sperm as it swims its way up the female reproductive tract in search of an ovum. The functions of testosterone. Testosterone has a number of effects on the male body. It controls the development, growth, and maintenance of the male sex organs. Just before birth, it causes the descent of the testes from the abdominal cavity into the scrotal sac. During puberty, it stimulates bone growth, resulting in the development of broad shoulders and narrow hips. It stimulates protein buildup in muscles, producing muscular development, typical of males with more bulk and firmness in their muscular physique. Testosterone stimulates maturation of sperm cells. It causes enlargement of the thyroid cartilage, resulting in the visible Adam's apple in males, and the deepening of the voice with its low range of pitch due to thick vocal cords. Other secondary male sex characteristics are also influenced by the production of testosterone. These include aggressive behavior and body hair patterns. Body hair patterns include the development of chest hair and axillary hair within hereditary limits, facial hair, and temporal hairline recession. The ducts of the system, as the sperm cells are formed, they are moved from the convoluted seminiferous tubules in the testis to the straight tubules at the tip of each lobule. Here the convoluted seminiferous tubules become linear and lose their convolutions. THSA lead to a network of ducts in the testis called the Reedy testis. The sperm are now transferred out of the testis through a series of coiled efferent ducts that empty into a single tube known as the ductus epididymis. The epididymis is a comma-shaped structure located along the posterior border of a testis, consisting principally of a tightly coiled tube called the ductus epididymis. The epididymis is the site where the sperm cells continue to mature. The tube is about 20 feet long and it takes the sperm about 20 days to move through this tube. The tube stores the maturing sperm cells while they develop their swimming capability via their flagella. The tube propels the sperm toward the next part of the system during ejaculation, when the smooth muscle of the wall of the tube contracts by peristalsis. The next part of the duct system is the ductus deferens, or the vas deferens. Within the tail of the epididymis, the ductus epididymis becomes less and less convoluted. At this point it is now referred to as the ductus or vas deferens. It is also commonly called the seminal duct. It is approximately 18 inches long and ascends the posterior border of the testis, penetrates the inguinal canal, and enters the pelvic cavity where it loops over the side and down the posterior surface of the urinary bladder. The tube is enclosed in a connective tissue sheath along with nerves and blood vessels. This sheath is called the spermatic cord. The end of the ductus or vas deferens has a dilated terminal portion known as the ampulla. Each ductus deferens empties into its ejaculatory duct, the next part of the system. When a vasectomy is performed for birth control, the physician performs this minor operation by making an incision into the scrotal sac to cut or cauterize the ductus or vas deferens. The testes still produce sperm, but they are now unable to make their way to the outside of the system. They eventually die and are reabsorbed by the body. A man becomes sterile by this procedure, but fluids are still produced and are ejaculated during intercourse. The male libido is not affected. Erections and ejaculations still occur, but there are no sperm in the semen. In addition, because testosterone is still being produced, all the secondary male sex characteristics are maintained. The next part of the tube is the ejaculatory duct. Posterior to the urinary bladder, each ductus deferens joins its ejaculatory duct. Each duct is about 1 inch long. The ducts eject spermatozoa into the urethra. The urethra is the terminal duct of the system. It serves as a common passageway for both spermatozoa coming from the testes and urine coming from the bladder. In the male, the urethra passes through the prostate gland, the urogenital diaphragm, and the penis. It is about 8 inches long and is subdivided into 3 parts: the prostatic urethra, which is surrounded by the prostate gland and is about 1 inch long; The membranous urethra about 1/2 inch long, which runs from the prostatic urethra to the penis, and the spongy or cavernous urethra, found within the penis and about 6 inches long, but vary according to the size of the penis. The spongy urethra enters the head or bulb of the penis and terminates at the male urethral orifice. The accessory glands. The accessory glands include the 2 seminal vesicles, the prostate gland, and the paired bulbourethral glands. These glands secrete the liquid portion of the semen, the sperm-containing fluid that is produced during ejaculation. The paired seminal vesicles are convoluted pouch-like structures approximately 2 inches in length. They are located posterior to and at the base of the urinary bladder in front of the rectum. They produce the alkaline, viscous component of semen that is rich in the sugar fructose and other nutrients for the sperm cells and pass it into the ejaculatory duct. They produce about 60% of the volume of semen. Because the duct of each seminal vesicle joins the ductus deferens on each side to form the ejaculatory duct, sperm and seminal fluid together enter the urethra during ejaculation. The prostate gland is a single, donut-shaped gland about the size and shape of a chestnut. It surrounds the superior portion, the prostatic urethra of the urethra just below the bladder. It also secretes an alkaline fluid that makes up about 13% to 33% of semen. Its fluid plays a role in activating the sperm cells to swim. The fluid enters the prostatic urethra through several small ducts. The prostate gland is located anterior to the rectum, and a physician can feel its size and texture by digital examination through the anterior wall of the rectum to check for an enlarged prostate, which could indicate cancer. The paired bulbourethral glands, also known as Cooper's glands, are about the size of peas. They are located beneath the prostate gland on either side of the membranous urethra. They secrete thick, viscous, alkaline mucus. Their ducts connect with the spongy urethra. This secretion is the first to move down the urethra when a man becomes sexually aroused and develops an erection. It functions as both a lubricant for sexual intercourse and as an agent to clean the urethra of any traces of acidic urine. Semen, also known as seminal fluid, is a mixture of sperm cells and the secretions of the seminal vesicles, the prostate, and the bulbourethral glands. The fluid is milky in color and sticky, due to the fructose sugar that provides the energy for the beating flagellum of each sperm cell. The semen is alkaline, with a pH of 7.2 to 7.6. This neutralizes the acidity of the female vagina and the male urethra and helps protect the sperm cell. The semen provides a transport medium for the swimming sperm cells. The average volume of semen per ejaculation is 2.5 to 6 milliliters, and the average range of spermatozoa ejaculated is 50 to 100 million/milliliters. If the number of spermatozoa falls below 20 million/milliliters, the man is considered to be sterile. Semen contains enzymes that activate sperm after ejaculation. The semen also contains an antibiotic called seminalplasmin, which has the capability of destroying certain bacteria. Because the female reproductive tract and the semen contain bacteria, the seminalplasmin helps keep these bacteria under control and thus helps protect the sperm and ensure fertilization. The penis is used to introduce or deliver spermatozoa into the female reproductive tract by being inserted into the vagina. The penis consists of a shaft whose distal end is a slightly enlarged region called the glans penis or head, which means shaped like an acorn. Covering the glans penis is a section of loose skin called the prepuce or foreskin. Occasionally, the foreskin is removed at birth by a surgical procedure called circumcision. This will be done if the foreskin does not pull back completely over the glans penis, resulting in future hygiene problems. If the skin is loose and pulls back, circumcision is not necessary. It is also done as a rite in certain religions. Internally, the penis is composed of 3 cylindrical masses of spongy tissue containing blood sinuses. During sexual stimulation, the arteries that supply the penis dilate, and large quantities of blood enter these blood sinuses. The expansion of these sinuses compresses the vein that would normally drain the penis so that most of this entry blood is retained. These changes in blood vessels produce an erection, which helps the penis penetrate the female vagina. Once sexual stimulation ceases, the arteries supplying the blood constrict and the veins drain the blood, resulting in the penis going limp and the end of the erection. During ejaculation, the smooth muscle sphincter at the base of the urinary bladder is closed. This ensures that urine is not expelled during ejaculation and that semen does not enter the urinary bladder.